Okay, everybody, welcome to the first colloquium. Usually I'm more man on it. Hello, everyone. Welcome to the first colloquium of 2026. So today we have Raj is coming from just across the, where are we? That way. Over there. He just joined the plant agros. Right, agro ecosystem sciences.
You can say pass.
It's not good either. He's a plant geneticist who started last year as part of the Raj program, was hired. And he comes to us from Purdue, where he did his PhD and then a postdoc with Brian Diltz's lab. And he does a lot of really clever and elegant genetics and also does something on the ligule sometimes, which some of you know is the most important part of the plant. I'm not seeing a lot of people who know this, but some of you know the ligule is the most. Will you talk about the ligule today?
I will.
It will briefly come up, and
you don't see the ligule there, but
well, you know that it's there. There's the ligule there, so that's the most important part. And has also done some great work helping to map. The first time I saw his name and heard about him was helping to map some classical maze mutants that had not been mapped for forever and kind of resolve some mysteries of it. So anyway, just really beautiful work, and I am looking forward to it. Always concerned. Thank you.
should I turn down the lights? Is it possible?
Or will cause light change.
All right. Perfect. Well, thank you so much for this opportunity. Let's see. Okay, so I'm a plant geneticist, as Jake mentioned. I think about the plant genetics a lot by combining the Mendelian world and the population genetics world. And I'll be touching upon some of these themes that I've been trying to hone over the last couple of years, mostly during my postdoc. And I'll briefly mention some of the things that we are doing in the lab, but I won't go too deep into that. So, so these two pictures here, I'll just mention a little bit. So this is one of the mutant, this leaf on the right. It's a dominant mutant. We call it Bellafleck 1. It's a autoimmune mutant, and the leaf on the left is a wild type sibling. And one of the things that you notice here are these are reddish rust pustules. So this is a biotrophic pathogen that is thriving on the wild type leaf, and you can see this mutant leaf is completely immune. So this is one of the mutant that we are studying. This mutant is resistant to all the biotrophic pathogens that we have tested so far. And the mutant on the right is a dominant mutant. It's a Ligulus mutant. We call it Ligulus 5. I'll briefly touch upon this one as well. And on the left is a wild type leaf shown on the side. And you can see the leaf sheath here. And this is the leaf blade. You can see a nice angle in a wild type leaf. But if you look at the mutant leaf, the leaf is very upright. And that's because it's missing a ligule, which you can't really see. It's on the inside. And then you can see the sheath is actually growing into the leaf blade. All right, so with this, I'll dive in. So first, I'll just briefly talk a little bit about the vision of the lab that I'm setting up here. So we have two students in the lab right now. One is working on corn, the other one is working on wheat. They're working on a bunch of mutants. They came out of various mutagenesis experiments, and they provide very broad spectrum disease resistance. And as I mentioned, this is just the mutant that was on the first slide here. And this mutant, I won't talk too much about this, but it's basically a glutamate receptor. We identified the gene. And so glutamate receptors are calcium channels. In this case, this one is localized to the plasma membrane, and it's constitutively pumping calcium into the cell. So we don't know exactly how this channel really integrates with the cell. So we're trying to understand that. So the other theme that the lab is working on is plant architecture. We are working on multiple mutants in corn, in sorghum, and wheat. So here is an example of a corn plant. So when I say plant architecture, I primarily mean height and leaf angle. So here's a corn plant. Here's the ear of the corn. So this the flag leaf height is the total height of the plant measured to the flag leaf, which is the last leaf, the terminal leaf on the plant. And then there's another height that really matters in corn, and that's the ear height. And that's because that's where all the seed is. So turns out the balance of ear height and the total height is really critical. And then I already mentioned about the leaf angle. Now, plant architecture doesn't just exist by itself. It also influences how it interacts with the microclimate and diseases. So I will briefly touch upon how some of these mutants, especially the narrow leaf angle mutants, are really susceptible to some of the diseases. And then finally, the lab is also interested in developing resources. So I'll talk a little bit about some of the things that we are doing along those lines. So here is a cover of one of the classical collection of maize mutants by Jerry Neufer, Ed Coe, and Sue Wessler, which has a collection of really beautiful pictures of a lot of mutants from maize. And then on the right here is a wheat assembly that we are working on. One of the graduates in the lab is working on right now, where we are trying to build reference quality assemblies for some of the wild species of wheat. And here we did a high throughput chromosome capture confirmation assay. And this is before we did the Hi-C. And then using Hi-C data, we corrected this assembly. And you can see these 7 chromosomes of this diploid species of wheat. So why do we care about plant architecture? Of course, we are interested in the molecular aspect. What are the genes? What are the pathways that are underlying some of these architectural traits? Some of the classical examples that really brought this to everyone's attention many decades ago is this example from wheat, where the DELA alleles, which are the genes in the GA signaling pathway, gibberellic acid signaling pathway. And then in rice, another example of using these SD1 mutants or semi-dwarf mutants, which is a mutation in the GA biosynthesis or the gibberellic acid biosynthesis. So this really kick-started the focus or attention of researchers towards using architecture, not just to understand the molecular aspect, but also use it in breeding. Then in maize, 2 other examples are the Ligulus 1 and Ligulus 2. So these are 2 loci that were selected upon by breeders many decades before we actually figured out what these genes are. And so they have been very heavily used in corn breeding. And then finally, there's a trade-off. So if you think about reducing height and protecting the plant from various abiotic stresses, it's going to influence the yield in a negative way. So you have to always find this optimum balance to get maximum productivity. So in the example of wheat, the RHT alleles were really revolutionary to reduce the height of the plant. But recently, we are learning that these plants are really susceptible to Fusarium head blight. So there's this pleiotropic effect that's driving some of this. So now there's a need to walk away from some of these DEL alleles that were really critical at one point, but now they're creating a new challenge. So Ligulus 1, which is one of the locusts that was selected for reducing the angle of the leaf, makes plants really susceptible to one of the fungal pathogen that causes northern cone leaf blight. So on the right is a graph that shows how the yield of corn really took off from 1930s to the 2000s. So the blue line here is the yield, and this is the axis here on the y-axis. You can see the yield. But the other thing you can also see is the number of plants that we grow per unit area also increased by quite a bit. And this was because of selecting on better plant architecture primarily reducing the leaf angle. So we were able to pack more plants without compromising photosynthesis. So the other thing that we know from the literature is that the genetic backgrounds, they're known to impact the severity of a lot of mutants. This is a classical example of the teopod mutants. So these are, so very recently, we have figured out the underlying gene for all of these. They're all microRNA 156 overexpressors. So here are the different variants of microRNA 156 that are highlighted on the right. So this mutant, if you have it in Ohio 51A inbred background, so this is one of the background of maize. If you have this mutant in this background, they're really severe. And here is the wild type plant for reference. But if you put the same mutants in MO17 background, or the phenotype completely disappears and the plant look pretty normal. So this is a really strong impact of the genetic background. So this is what really drove me to study these natural modifiers that are impacting these mutants, and I've been chasing these through various projects over the years. So one of the cool way that you can study dominant mutants is by simply taking the mutant of interest as a heterozygote and crossing it with different inbred lines. And because it's dominant, you will be able to see the phenotype in the F1 family. So in this case, we have an example of a dominant dwarf mutant, which we cross with, let's say, an inbred A. And in the F1, you get 1:1 segregation. And in this case, you can see that the mutant is slightly shorter than the isogenic wild type sibling. But in this second cross, where you cross it with inbred B, the mutants are really severe. So this is very similar to this example here. So the advantage of this F1 hybrid is you can cross this mutant to different populations and use the variation across these families to map the loci that are driving these modifications. So one of the advantage of this is you have this really nice case control design, because these two siblings are isogenic. They're only differing at this mutant locus. So you can detect these modifiers by comparing different F1 hybrids, and you can do a genome-wide association mapping. So the disadvantage of this screen is sometimes you miss a lot of recessive modifiers. So you're more likely going to detect dominant modifiers in the screen, and will miss some of the recessive modifiers. And also you'll miss some of the rare alleles, especially if you're using an association mapping population with where you have tons and tons of backgrounds that you're inheriting. But you can actually get around that if you use certain mapping populations. And then the last disadvantage is you have to make hundreds of crosses. But in corn, making crosses is not that hard. So that's why we have been using corn to really move this project forward. So with this, here's my outline for the talk. So initially, I'll touch upon the concept. So this is the paper that I shared with you. So I will touch upon that, but I'll also mention some of the papers that came before that led to that specific project. So this is really the story about the chlorophyll biosynthetic mutant. I'm also going to talk about the application of this concept at the end of the talk. But before that, I'll touch upon some of the genetic tools that we are developing. And I also shared a publication on that with you guys. So let's talk about the chlorophyll biosynthetic mutants, where we're using a dominant mutant to characterize modifiers. So this project really started with this chlorophyll biosynthetic mutant shown here called the OLL1N1989 allele. So this is a semi-dominant allele. It has a leucine to phenylalanine substitution. And this particular allele, it creates a competitive inhibitory allele to the magnesium chelatase complex. So magnesium chelatase is the first enzyme that commits protoporphyrin 9 to chlorophyll biosynthesis. So this is the first enzyme in that pathway. And this enzyme is a multimer, so it has 3 different subunits. And subunit I is encoded by this OLL1. So when you have this particular mutant allele, it poisons the complex, and you no longer get magnesium protoporphyrin 9 production. So this mutant, because it has this really striking pale green phenotype as a heterozygote, is a really good reporter for looking at any modifier using this simple phenotype. So we use this as a reporter, and the design that was carried out was we basically crossed this mutant as a heterozygote. So this is this allele is on this locus is on chromosome 10. So I'm just showing you chromosome 10 here, and you can see it's a heterozygote. So this black dot represents the mutant allele. So this mutant was crossed as a heterozygote to 378 different inbred lines from the Maze Association panel. So this is just a panel that consists of different lines that came from around the world. So they're very different from each other. And these are inbred. So they're highly, highly homozygous. So when you cross this mutant to these inbred lines, you get F1 families. And in each F1 family, you'll have 2 kinds of genotypes. You'll have the wild-type siblings, and then you'll have the mutant siblings. So we made this population, planted it out in the field, and we phenotyped, we measured the chlorophyll content by using a small instrument that you can just put on the leaf, and it will give you a number, how green the leaf is. And then we also measured plant height, flowering time, and stalk width. I'm not going to talk a lot about these, but I'll talk about the chlorophyll. So once we had the data, we simply took the genotypic data from the inbreds, which we already had, and we simply carried out genome-wide association mapping. So here is the Manhattan plot, which basically shows the association of SNPs with different phenotypes. So the x-axis are 10 chromosomes of maize, and each dot here represents the SNP. And each color here represents the different phenotype that we collected. And the y-axis is the p-value. So higher you are on the y-axis, more significant you are. So I really want you to focus on this really significant association on chromosome 10. And we call this QTL very oil yellow 1, because this is the location that makes plants extremely yellow. So we wanted to study what's happening at this locus. So it turns out that this locus harbors the oil yellow 1 gene itself. So think about this. We started with the mutant allele at oil yellow 1, and we map it back to oil yellow 1. So what's going on? So we carried out multiple experiments to test if it's the expression or it's the protein that's driving this phenotype. So here's the expression association mapping. So we took the expression data from the same population that we crossed. We took the expression data for oil yellow 1 from 2 different tissues. So it's the expression data from leaves at maturity during the day and leaves at maturity during night. And what you see here is a strong association of the SNPs with the expression data during both day as well as night. And what's shown in the panel B here are different SNPs in the 5' and the 3' end of this gene. And you can see that the SNPs in the night tissue are increasing the expression of the gene. And the SNPs on the 5' end in the day tissue are decreasing the expression of the gene. And if you look at the chlorophyll phenotype, it's moving hand in hand. So as you change the expression of the gene, as you increase the expression of the wild-type allele, you make the plants more greener. As you turn down the expression of the wild-type allele, you make the mutants more pale. So with this, I'll just introduce the model of this phenomena. So basically, in the suppressing backgrounds, we have plants that are making the same amount of mutant mRNA, but the wild-type allele is what's really driving the phenotype. So in the suppressing backgrounds, you have equal expression of the wild-type as well as the mutant allele. But in the enhancing backgrounds, you actually have a much lower expression of just the wild type allele. And that puts more pressure on the production of some of the intermediates in the pathway. So if you are more visual, like I am, so this is what the phenotype looks like in the field. So here we have the mutant in a near isogenic background. So both of the backgrounds are identical except the wild type allele. So here we have MO17 crossed to this mutant in B73. And in this case, the wild type allele is coming from MO17. And you can see that these yellow plants are extremely severe, and the wild type siblings are really green. But if we swap the wild type allele in MO17 with the suppressing allele from B73, you can no longer distinguish the mutants from the wild type. So this impact on chlorophyll also impacts other processes, for example, tillering. So here is a classical mutant, teosinte branch 1, along with the mutant allele, with the suppressing allele, and with the enhancing allele. And you can see that the tillering has reduced significantly. And this is just a quantification of this data. Just like that, there's another classical mutant, grassy tiller 1, which is downstream of teosinte branch 1. We see the same thing. As you incorporate the enhancing allele, you basically suppress tillering in the grassy tiller 1 mutant. So one of the puzzling phenotype that we observed during this experiment is the impact on plant height, and we still don't have any molecular explanation for this phenotype. So the observation is the following. So here is the wild type plant on the left. In the middle, we have a mutant plant with a suppressing allele. And here is the mutant plant on the right with the enhancing allele. So the mutant plants that have the suppressing allele tend to be taller than the wild type plants. And the mutant plants that are more severe tends to be shorter. This phenotype is easy to explain. You make less chlorophyll, you're shorter. But how come you can be taller when you have less chlorophyll? So this was really apparent when we actually plotted the data. So on the x-axis, we have the chlorophyll of the mutant, and the y-axis is the difference in height between the wild-type sibling and the mutant sibling. If you are at the 0 line, there's no difference between the mutant and wild-type. So as you can see, if the mutants have higher chlorophyll, they tend to be taller than the wild-type siblings. But as this chlorophyll level drops, you see a sharp drop in plant height as well. So here is the data expressed as a quartile. So you can see the bottom quartile sees a big drop in chlorophyll and also in height. So we hypothesized that this is probably because of the phytochromes. We measured them. We don't see any difference. We looked at different light regimes, far red light. We still don't see any difference. So we still are exploring what's driving this phenomena, and this has not been reported with other mutants before this one. So just to summarize the first part of the talk, so we observe some of the things that I haven't shown because of the lack of time. So we see reduce in tassel branching as well. So tassel is the inflorescence, the top inflorescence on the corn plant. So we noticed that the tassel branching is reduced significantly, and it's reduced in an additive fashion if you have the suppressing allele versus the enhancing allele. We also observe suppression in senescence. So if you prevent pollination in maize, you will see the leaves will just senesce like crazy because they just accumulate a lot of sugars, and the sugars have nowhere to go. So the leaves will just start to senesce. But if you have these alleles in the background, you can modify the phenotype. So the other thing we also observe is that the natural alleles at way 1, the expression, the cis-acting expression polymorphism, they are driving the porphyrin pathway by regulating them in trans. And this is happening also in the wild type plants. So we initially discovered this mechanism in the mutants, but we see a very weak transcriptional response even in the wild type plants. So altogether, there's a coordinated transcriptional regulation that's happening in the wild type plants, which was really brought to attention by observing the mutants. And this is very similar to some of the classical mutants that people have identified to characterize some of the other pathways. Ligulus 1 and Ligulus 2 are a good example of that. Those two genes were identified by mutants that had a very strong phenotype. But if you go and study the natural population, you see the impact of those alleles in the natural population as well. All right, so with this, I'll go to the second part of the talk, where I'll discuss about some of the genetic tools that we are developing. So the first thing I'll talk about is the use of somatic chimeras that we're using to eliminate fine mapping. So many of you in the room are familiar with linkage mapping, and you're using them for your research. You know, if you're working with a Mendelian trait, you're trying to narrow down the gene to a small interval. It could be a really hard task, especially when you're fine mapping close to the centromere. So we thought about if we can reduce the workload. And this idea really was born out of working with the maize lesion mutants. So I showed you maize lesion mutant in the beginning, the Belaflect 1 at the beginning of my talk. So the hypothesis was that if we can take the heterozygous seed of this mutant and treat it with EMS, we can potentially knock out the dominant allele, and we should be able to get a wild-type phenotype on an otherwise mutant plant. So we carried out this experiment, and we got these two independent chimeras in a very small population. So here, both of these plants are Bella mutant plants. You can see this phenotype with lesions on this sector here, but then you also see this really nice wild-type phenotype. So we hypothesized that this is because we have knocked out the dominant allele. So we carried out whole genome sequencing on these two independent tissues. And the hypothesis is because EMS is causing random mutations, these are going to be two independent mutations in the same allele. I'm not going to show you the data, but basically what we found is there was one gene, it's a glutamate receptor. It actually had a missense mutation in the Bella allele, but in these two sectors, there were two other missense mutations in these two sectors. So that helped us identify the gene in a big window. So when I was working on this project, I wondered if we could use this approach to characterize other mutants, other developmental mutants. So one of the mutants that really stood out was this male sterile 8. So we had a recessive allele in the lab. This is in sorghum. So this is a wild-type inflorescence of sorghum plant, and you can see these fresh yellow anthers that are full of pollen sticking out, and there is a homozygous mutant which has white anthers. And you can see this phenotype more in this close-up. So you have these feathery stigmas and these three yellow and plump anthers in the wild type, and in the mutant, these anthers are white and they're flat. And if you look at the micrograph here in the lobes of the anthers, you can see they're full of microspores or pollen grains, and the mutants don't have any pollen grains. And here's the anatomy of the pollen of the anthers. So you can see that the tapetum layer is very much intact in the wild type individuals, but in the mutants, the tapetum layer is completely disintegrated. So this mutant, we didn't know what the gene is. We only had one allele. So one other thing that you do to confirm the gene is you map the gene, map the region, and then you identify an independent allele. So we did a bulk segment analysis, and we mapped it to chromosome 4. And now to get the second allele, we simply made an F1 material by crossing this mutant with wild-type pollen, and then we just mutagenized this with EMS. And the idea was, if we knock out the wild-type allele, in this case, we will see a sector, a chimera, just like I showed you on the leaf, but in this case, we'll see a chimera in the inflorescence, which will have the sterile sector. So all the F1s are expected to be fertile because the mutant allele is recessive. So if you look at this inflorescence closely, you see these yellow anthers over here, but if you stare in the middle, the anthers are white. And in this picture over here, we simply excised some of these florets on the edges to separate this tissue from the rest of the plant. And then I simply carried out whole genome sequencing on some of the tissue from the fertile florets and some of the tissue from the sterile florets. And what we found was there was one gene, beta HLH, a transcription factor, that actually had a nonsense mutation, a stop codon in the fertile florets, but in the sterile florets, we had another mutation, a nonsense mutation on the wild-type allele. And these sectors, this sector was able to produce seed. So in this case, we actually were able to recover this allele germinally. So again, so this is a classical complementation test, basically. So using the screen to look for a second allele by using this targeted design. And if you look at the DNA sequence at this locus, you can see that these are the reads spanning across the region of this beta HLH gene in the fertile tissue, where you have this causative SNP that causes a stop codon, Q150. And in the sterile tissue, we have this other, we actually have two mutations in this case. We had a missense mutation as well as stop codon that were linked in cis. So one of the things that's really interesting to observe here is because these two alleles were so close to each other, we were actually able to see reads that were able to separate these two alleles and make them more stranded. So for example, if you follow this read that had this original reference mutation that was present in MS8-1, those reads don't have this mutation. So you know it's happening on one of the molecule. But the reads that had this new mutation are missing this reference mutation. So you know that these two alleles are they're independent from each other. So this is a confirmation at the molecular level, so for the complementation test. So in conclusion, MS8 is encoded by a beta HLH 122 in sorghum. So this MS8-2 is a novel mutant allele that was derived from this particular background. So one of the advantage of using this screen is that you can derive a new allele in any background. You don't need to use a certain background like you would for, let's say, gene editing, where certain backgrounds are more suitable for transformation. So I noticed some of you asked the question that were passed on to me from Claire, how this method is better, or how does it compare to gene editing? So the advantage of this method is you don't need to know what the gene is. You're letting the screen do the job for you. But if you're doing gene editing, you need to know the target that you're manipulating. So it's also a great tool for traits where the phenotype is cell autonomous. So you can use it for disease resistance. I talked about the example of the lesion mutant, but you can also use it for inflorescence architecture where the phenotype could be discerned on the plant body itself. So when I was doing the sorghum experiment, I also observed a bunch of these other really cool sectors. These are potentially other dominant mutants. If you look at the first figure here, you can see this really nice sector, which shows up as almost necrosis on the leaf. But then this sector, it gives rise to these red kernels. on the inflorescence. And then here's another sector where you can see this albino sector on the leaf. It's present on multiple leaves, so you can see it over here. You can also see it over here. But then you see this albino sector that also goes into the inflorescence. So you can recover all sorts of these other cool dominant alleles when you're doing these kind of experiments. Okay, so with this, I'll jump into the next topic of developing these new genetic tools. Just sticking to the theme of working with Mendelian traits. So one of the questions that we are always running into is when we find these mutants, where do they map? That's pretty common. So in order to facilitate genotyping and facilitate genetic studies, we came up with this new method where we can map mutants for pretty cheap. So in this case, there was a sequencing service at Purdue. We actually have a similar service here at the Biotech Center where you can get long reads for $15 per sample. You can generate random reads. You can use it for all sorts of tricks. But that service gives you a couple thousand reads, and you can actually map mutants with those reads. So that's exactly what we did here. And the motivation behind this project was we had this dominant allele that came from one of the mutant screen. This came as from an EMS population in MO17 background. It's an inbred background in maize. And the first hypothesis we had was that this mutant looks very similar to LIG3 and LIG4, which are two loci in maize. And there are multiple dominant alleles that have been characterized for LIG3 and LIG4. So one of the problems with working with dominant mutants is you can't really do a complementation test like you could for a loss of function allele, or at least they are hard to interpret. So the easiest way to figure that out would be simply map it and see if it maps to LIG3 or LIG4 genomic position. So LIG1 is on chromosome 2, LIG2 is on chromosome 3, LIG3 is on 3, and LIG4 is on 8. So we decided to map this mutant. So for this, we generated a BC1 F1 population. So just a reminder, this mutant originally came from MO17. So we crossed it to B73 twice and made this BC1 F1 population. So we took the pool from BC1 F1 and simply did a short read sequencing where we were able to generate 100,000 reads through this service at Purdue, which cost $21 per sample. And the coverage of this 100,000 read is in the order of 0.01x. So it's really small. So how do you use this data in any meaningful way? So we came up with this pipeline. So you align the reads from the sample to your reference genome. In this case, the reference was B73. And then you simply take all the polymorphic SNPs between the two parents that you're using. So in this case, it was B73 and MO17. And then you simply assign these reads to whether they're reference reads or the non-reference reads, whether they're B73 or MO17. Now, in this case, the first read here does not fall on any SNP. So this is a non-informative read. So this read is informative because it overlaps with the SNP. So you can't really use this individual read in any meaningful way because at any given spot, you only have a single read. So either you're going to be homozygous for one parent or the other, or you're going to be heterozygous. But if you take an average across a big region, you can predict the genotype in that particular stretch of DNA. So that's what we did. And in this case, you can see that there are two reads here that are MO17, and there's a single read here. There's only one informative read, which is B73. So we can say that in this chunk of sequence, it's 66% MO17 or non-reference. So we did this for the two pools that we sequenced. So in the wild-type pool, we observed this spot on chromosome 6, which was 100% B73. And each dot here is a bin of 5 MB. So we're looking at the allele frequency across 5 MB chunk, and we repeat this exercise across the whole genome. So in the mutant pool, which is heterozygous, we observe this depression of allele frequency at the same spot on chromosome 6. And if you take the ratios, you can see that the signal is extremely clear. So in this case, this locus mapped to chromosome 6, where we have no known ligulous mutant. So therefore, we were able to call it a Lig5 locus, and this allele is Lig5-1. So you can use this design to do all sorts of things. You can do haplotype analysis. So if you have a pedigreed population, you can use to figure out what background they came from. You can genotype RILs, NILs, identify breakpoints. It's also great for teaching because you can do this analysis on a regular laptop. It only takes like 10 minutes because you have so little data. So the other thing we observed in this mutant is really susceptible to corn smut. So here is this mutant in 3 different hybrid backgrounds, and you can see this fruiting body from Osteolago matus, which is a biotrophic pathogen that causes corn smut. It loves this mutant. So, we don't know what's driving this interaction, but this is not the first time we have seen a ligulous mutant showing some susceptibility to a fungal pathogen. So in conclusion, this approach is extremely cheap. It's effective for background selection. So if you're backcrossing something and you want to chase a certain QTL, it's great for those kind of exercises. You can use it to eliminate contaminants if you have using an enhancer suppressor screen. I have some slides later on, I can show it to you. You can do some haplotype determination in a population. You can also use it for genotyping a RIL or a DH or a double haploid population. So we have a collaboration going on right now in Barley with the folks at the University of Alberta. And I'm actually giving a talk next week, same room next Wednesday at noon. So if you want to listen to what we're doing there, feel free to check it out. So yeah, I have a couple more slides. I noticed I'm at 50-minute mark. Can I? Okay. So finally, I want to end. I have 3 slides. So I want to show you 2 examples. So I talked about this concept of using Mendelian mutants and combining it with the population genetics. I want to give you 2 examples where we were able to use this to find some really interesting mechanisms that were driving 2 different phenotypes. So the first example is a case of mutator suppressibility. So mutator is a DNA transposon, and we were actually able to use one of the allele that shows this mutator suppressibility to find a transposase element. So this is a really busy slide, so bear with me. So the panel 1 here shows the mutant that we use. So we use this mutant called les28-1. So there were 2 different alleles, les28-1, les28-2. This mutation is in the enzyme in the porphyrin pathway. So the enzyme is uroporphyrin synthase. This enzyme is also present in humans or animals. So both of these alleles are mutator insertions. So this is a DNA transposon. -1 allele has an insertion in the first intron. -2 has an insertion in the 5' UTR. -1 is slightly more severe, as shown here. It has these small lesions, and if you cover these leaves with the aluminum foil protected from light, you don't get any lesions. So these are really triggered by light. So les28-2, it does have lesions. It's much weaker in a heterozygous. It's more severe as a homozygote. So one of the things that we observed with this allele is it's actually really sensitive to the presence of transposase enzyme in the background, which is really shown in this panel 2 here. So we cross this mutant. So these 3 leaves here are mutant leaves. So this is les28-2, and this is the wild type leaf here. So the wild type leaf does not have any lesions. So the mutant, if you have transposase in the background, you get lesions. But if you epigenetically silence this element that produces transposase, you actually suppress lesions. And that's what's shown here in this graph here. So these 3 they're all in the same order. So if you have this element called mu killer, which actually eliminates all the transposase in the background, you get very little lesions. So we knew this mutant is sensitive to having transposase in the background. So one of the questions we asked is, can we identify any modifiers of this mutant? Can we identify natural variation in transposability in the backgrounds of maize? So again, we crossed this mutant with the association panel, the same design that I showed you before, and we simply scored the lesion severity of mutants in these different F1 families. So we scored them from 0 to 5, with 0 being no lesions, 5 with severe lesions, carried out genome-wide association, found this really strong QTL on chromosome 4. So what is this QTL? Turns out it was a big region, 500 kb. There was no protein coding gene that was annotated in this window. And when we looked at if there's anything expressed, we found an element that was very similar to the transposase itself. So it was a sequence that was very similar to transposase. And this transposase element was slightly different than the DNA transposase that interacts with the mu elements. So this element had a different domains that we expect to be present in a typical transposase, including the nuclear localization signal that allows it to enter the nucleus. So when we were looking at different lines in the maize association panel, we found multiple alleles. So some of the lines actually had an insertion of 131 base pair sequence that created a premature stop codon. And this mutation eliminated the nuclear localization signal. And we found a bunch of other mutations that caused premature stop codon. And the interesting thing about this particular haplotype was all the lines that actually had a functional allele were tagged with one SNP, because initially we are using SNPs to map these, right? So we have a biallelic SNP, but we have multiple alleles at this locus. So we found that the functional allele of this transposase was linked with one of the SNP. And then a lot of these other non-functional were tagged by the other SNP. So we basically had a tag SNP that was able to report the functional state of this locus. And the other interesting thing we found about this locus was that this is polymorphic in domesticated maize. But this locus was exapted from a historical transposase long time ago. It actually happened before the split of Trypsicum from the Zea lineage, which is more than 650,000 years ago. So we think that this is a sequence, it's a transposase that has undergone multiple changes over time. And now it's actually acting as a proper gene. And it doesn't have TIR sequences. There's nothing that looks remotely like a transposable element. So it's actually fixed in place. And but if we go beyond Trypsicum, we don't see this particular gene in the syntenic position. So finally, I'll talk, I only have one slide here. So this is a story that just came out last month, actually, right after I sent those 2 publications to you guys. So we recently characterized a mutant we call dwarf 13. It's also a glutamate receptor. This particular mutant, it's a dominant allele called D13-1. It actually reduces the height of the plant in certain backgrounds. So again, it gets modified by backgrounds. So you can guess what we did. We actually crossed it with the association panel, and we found 2 modifiers. One of them was a cis-acting expression polymorphism at the D13 locus itself. So this is very similar to what I mentioned about oil yellow. But then we also found another modifier, which actually encoded a Cornichon homolog. So Cornichons, they're involved in trafficking some of these glutamate receptors. It has been characterized with some of these animal receptors. And we think we have identified one of the interacting partner for D13, and it's Cornichon. So with this, I will conclude. So you can combine the Mendelian and the population genetics using this very general approach you can link known expression polymorphisms that exist in the wild-type population to causality, and you can start to build some of these interesting mechanisms that are driving some of these phenotypes. So we have identified a link between growth and development. It's a lot of that work is not that surprising, but it's an interesting way to study them, I believe. And then we also identified some of the phenotypes, for example, the plant height, where I showed you that you can increase the height by even dropping the chlorophyll a little bit. So we still don't have an explanation for this, and we observe this over and over. At this point, we have measured, I would say, easily over 10,000 plants, and this phenotype is so repeatable in every single population we study. This approach has also facilitated discovery of novel mechanism of transposon regulation, like the example that I talked about for LUS28. And then finally, when I talk about D13, a glutamate receptor, we can identify how it's regulated by different mechanisms in maize in the natural population. So with this, I would like to acknowledge mostly my postdoc advisor who really put me on this trajectory, and it has been quite interesting, and we have learned a lot. And now we're, taking it in different directions, also taking it to different species, especially wheat and sorghum. And then members of the lab, Aman, postdoc there, Tina, who was really helpful when I got started there. And then I would like to thank all the collaborators. I didn't get to talk about the phase change work that we did with Scott. And then the all yellow work was done in collaboration with David Braun at University of Missouri. The Belaflect 1 work will hopefully come out in the next couple of months. So really thankful to Peter and Sid Buell. Andrea Eveland, we worked on the MS8 project. We were actually racing with Andrea because she was in the process. They thought they knew the gene, so they were doing their CRISPR. And we did this EMS screen. It took us a month, and we got the gene before them. So we beat them. And then I would like to thank new collaborators, some of the barley work that we are doing, and then folks, different facilities that were really part of this incredible journey, and all the funding from different agencies. And with this, I'll be happy to answer any questions. Thank you.
I'm so glad your message for questions.
I was wondering, and my question is about the somatic engineering, like this. How applicable do you think that is for polygenic traits?
Don't do it. Yeah, so I would suggest if you have a phenotype which is cell autonomous, and then also if it's monogenic. Now, if you have a polygenic trait and it's controlled by, I'm assuming it's controlled by multiple QTLs, and you have Mendelized those QTLs, you can try it. And that's something we have thought about. If you have, because usually when people say polygenic, usually there are 2 or 3 QTLs that are really running the show. So if you can Mendelize those, fix those QTLs, and the phenotype is easy to observe on the plant body, I think you can do it. But don't try it in a segregating population. Yeah. I'm sorry?
I was asking whether you did line by tester for your experiment design. And then if that is the case, why you do that, why you chose that over maybe full dialogue? Because I'm thinking that dialogue will give you some DSK effect and SK effect that will help you to know whether this genes as an additive contribution, for example. Maybe even some matching effects, and probably might even increase your parts and that's, the QC of them present in the background.
Yeah, I agree with you, actually. Yes, I think if you want to identify a lot of these modifiers, especially with additive inheritance. Yes, there are other populations that you can use. In this case, it was really the availability of resources. We had this mapping population, and also we were interested in doing an untargeted experiment where where maybe we have a potential to observe some of the phenotypes that we don't even know exist. And we actually had quite a few of those. Some of these populations, I didn't get to talk about Bella, but that mutant, we actually didn't know is resistant to disease. The only way we were actually able to study that is because if the mutant is resistant, you need to have it in a susceptible background. If it's in a resistant background, you don't know if it's resistant, right? So we were actually able to use some of these populations, which are so diverse in phenotype, to really find these make these interesting observations that we can then follow up on. So, but you're right. I think there's a lot of fine tuning that you can do from the population side, where you can use a dial, you can use a magic population, you can use a NAM design, and yeah, and those will be more powerful. So we're definitely missing a lot.
Regarding synthesis, I mean, it's really cool to see these phenotypes being expressed like light, yellow, and deep green colors.
Yeah.
But I was wondering whether maybe thinking of or you studied something about how this the reduction in focus in the chlorophyll one affects the photosynthesis systems, for example, the system 1, system 2, or in the highlight or membranes. If possibly, whether it has an effect on certain genes that are probably down spread.
Yeah, so we actually published a paper where we did a pretty deep dive on the physiology and photosynthesis. We looked at the sugar and starches, but also we measured photosynthesis rate. In that case, I think we just did a light core, so we didn't really get into photosystem 1 and 2. Because it's a chlorophyll mutant, we think it's going to be consequential mostly to overall photosynthesis, probably through both. But we didn't really tease apart those mechanisms. We are actually, yeah, I'll just stop there. We try to do something, but then yeah, we didn't go further.
Following up on that, you.
Uh-huh.
You sprinkled for that. You said, "Oh, I think maybe phytochromes is involved in the height," but you didn't say anything about why you think phytochromes are involved in it. Did you? I mean, I could think of 20 different reasons. So yeah, tell me.
Well, the reason, because the chlorophyll pathway bifurcates, it feeds into the production of phytochromes, so into the bilins. So there was a mutant, elongated mesocaudal 1, that was characterized by Rory, and they found that mutant is pale and is taller, right? So those mutants are tall, because they're trying to make up for reduced phytochrome. So that's why we were thinking it's probably because, these are the pathways they're sharing the precursor. You're probably changing the flux in the pathway. So maybe you're reducing, or there's some feedback regulation going on. We actually made a double mutant between ELM1 and oil yellow. They were synergistic. So they got more really severe. So there was a synergistic effect, and they were much shorter. So we couldn't really test it. We also did a light experiment where we looked at the mesocaudal elongation pretty early on by inducing the phytochromes by using far red light. We didn't see any difference. So the only thing I guess we didn't do is actually measure the bilins themselves.
You have to cross the phytochrome itself.
No, we only crossed ELM1. We didn't cross the phi
But so
mutants.
I guess the reason I'm drawn to is if you would therefore say there maybe is a little bit of a shade avoidance response. Something
Yes.
we're seeing,
Yes.
I'll just say for those of you who don't know, in May's world, there's a big
Mhm.
hard rail that I talk about. So okay, cool. But I like. Can I just
No, it's okay.
Was it about the defense trade-off in your legal five something? Because was it at the beginning that you mentioned that they're shorter, but they have higher yield? Or was that a different?
So that was in context to just in general that height and yield, they go hand in hand. If you reduce the height by too much, you're going to reduce the biomass and the harvest index, the plants is just going to yield less. So I think that was more in context to that. But then also there's an interaction between height and disease in wheat. So the RHT alleles, the del alleles in wheat, they make it more susceptible to fusarium head blight. And yeah.
Rows that you think are trading off with disease resistance, because it's not just like all growth is trading off.
Yeah, I can't tell you much really. But yes, growth and defense. You guys study quite a bit. I would say one thing about the Lig5, because you have this mutant actually does have a lot of it doesn't have knots, but it actually does have, it looks like it has higher cell division happening near the leaf collar. So one of the very simplistic hypotheses, like, you have these meristematic growth happening, cells are dividing, so they're not maturing. So maybe there's less lignification, other things that it's just,
Is the infection ever happening?
it's happening.
Like the ligaments not there, that it's not protecting this site from infection. Is that?
It's happening at the collar. Yeah. Yeah. In every case, like, yeah, it's very consistent in that regard. It's always at the collar.
Okay, we'll let you guys that you can go meet with the students in the session. So okay, let's thank Raj again.
Thank you.